Hi everyone, welcome back to the next video of the course. So we'll keep moving ahead and understand in this video how diversity is generated in a prokaryotic population where reproduction is asexual. So first thing we should realize is that this we have an individual which grows under appropriate conditions, and this individual then divides to give two individuals. And this type of inheritance where one individual has given two is called uniparental inheritance. Uniparental inheritance, because what an individual has acquired has only come from one parent and not two parents, such as the case with complex organisms such as human beings. So in such a context, what we were discussing in the last video was that if we have a population, microbial population, where at t equal to 0, the number of individuals is, let's say, equal to n naught, and we have this population in a growth flask which contains appropriate nutrients to support cell growth and cell division. If we keep this population under these conditions and we let some time pass on, then at a future time t equal to t naught, the number of individuals would have increased to, let's say, nt. And we need to understand a few properties associated with this process. So the first one, we looked at this last time also, is how many divisions took place in this time. And that number is simply equal to, so number of divisions in t naught time is simply equal to nt minus n naught. And that's equal to this number of divisions is equal to this expression because of this observation that every cell division increases count of the population by exactly 1. So one cell division implies increase in population by exactly 1. Hence the increase in population that we see here in the case that we are looking at is simply nt minus n naught, and hence the number of divisions that need to take place to facilitate an increase from nt to reach n naught is simply equal to nt minus n naught. That is the first thing that we note about growth taking place in this flask. The second thing that we want to note is that how many generations passed in this time. How many generations passed in t naught time. And to calculate that, what we should realize is that, let us do this on the next slide, that we started at t equal to 0, the number was n naught, and we let growth take place for a while, t is equal to t naught, and number of individuals in the flask is nt. So this transition has happened in this time, and we want to understand that we now know that number of cell divisions in this process that took place over t naught time is simply equal to number of cells at the end minus number of cells at the beginning of the process. And what we are interested in is how many number of generations happened in this time. So one way to think about this is that at start the number of individuals was simply equal to n naught. after one generation, number of individuals. So, after one generation, what has happened is that if there were n naught cells, after one generation, every one of these cells has divided exactly once. So, instead of n naught, we have 2 times n naught cells after exactly one generation. So, after one generation, the number of individuals in the flask is going to be 2 times n naught. Similarly, let us do one more, that after 2 generations, the number of individuals will be 2 times n naught, which is 4 n naught, or simply 2 to the power 2 n naught. Because now each one of these, so we started with n naught, after one generation we had 2 n naught, and now after another generation every one of these 2 n naught individuals is going to divide, and we are going to end up with 4 n naught individuals. What you should realize at this point is that if we keep doing this process over and over again, we will get this relationship that what happens, the number of cells after k generations is simply equal to number of individuals after k generations is simply equal to 2 to the power k n naught. Because for every generation we simply multiply the number of existing individuals by 2, because in one generation every individual is dividing exactly once. Hence, at the end of k generations, we have this many number of individuals. And obviously, in the scenario that we are discussing here, it's the value of k that we are interested in, that what would be the number of generations that took place which increased the population size from n naught to nt. And to find that value of k, we simply have to equate these two numbers, and we get 2 to the power k times n naught is equal to nt. So, we solve this, since nt is given to us, n naught is known to us. What we have to do is, for this, what value of k is this equation true for? And we do this simply by taking log, and we get, we can take n naught to the other side, we get 2 to the power k is equal to nt by n naught, or if we take log to the base 2, k is simply equal to ln base 2 of nt by n naught. That is the number of generations that happened while this expansion of population took place, as the population changed from size n naught to a size nt. What that means is that if a population increased from, let us say, size 100 to size 400, then number of generations k is equal to ln of to the base 2 of 400 to 100, which is simply ln base 2 of 4, which is just ln base 2 to the power square. The 2 comes outside, and then ln of base 2 is simply 1. So, this is just 2 generations. So, this gives us mathematically, but we can simply see here that if you were to think of 100 and 400, after one generation everybody divides and this becomes 200. So, we have 200 individuals, and in the very next generation each one of these 200 individuals divides and we get to a population size 400. So, this is generation number 1, generation number 2. So, exactly 2 generations is what it takes for this population to become 4 times. And we can keep on doing these experiments of keep on studying these numbers of the kind to understand how many generations did it take place. So, that's about how many generations was it, how many generations did it take for the expansion of the population to take place, or how many cell divisions happened, and so on and so forth. One key thing that needs to happen for cell division to take place is that if you think of an E. coli, while we have been looking at the external size and number of the bacteria, something needs to happen internally inside the bacteria before it's ready for before it's ready for division. And one of the key requirements of cell division is that the DNA must be copied. So, let us imagine that this is an E. coli cell, this is its DNA, and after division this cell is going to give me 2 individuals, but each one of these individuals is also going to carry the DNA. So, what that means is that as part of the growth process, of growth, DNA must be copied. And that makes sense, and that way there are 2 copies of DNA and each of the 2 progenies inherits one such copy. E. coli's genome that needs to be copied is different strains of E. coli have different lengths, but it's 4 to 5 into 10 power 6 nucleotides. Division is not division, replication of DNA is done by a machine called DNA polymerase. It should strike you immediately that the process of transcription was done by a machine called RNA polymerase. It was a machine that synthesized the polymer called RNA. DNA polymerase is a machine that is responsible for synthesis of a polymer called DNA. So, division is done by DNA polymerase. Now, since this is essentially a cellular machine, and no machine in any context works with 100% efficiency. In the context of replication of DNA, what that means is that as the DNA of an individual is copied into 2, as the cell prepares for cell division such that each of the 2 progenies inherits 1 copy of the DNA, this replication process will not be 100% accurate, and some errors will be made in the process of replication of DNA. What that means is that we are talking of literally millions of nucleotides like A, T, G, G and so on and so forth. This is circular DNA. So, it circles back onto itself, and for cell division to take place, all of this has to be copied. So, let us say the copy will also be A, T, G, G and so on and so forth, and each of the 2 progenies gets a copy. However, with some rate, this machine that is responsible for copying the DNA and making the additional copy makes errors, and those errors can be of different types. But to take an example, what might happen is that while DNA is being copied, instead of a second G here, it inserts a C. What that means, and let us say this is the only error that takes place in the entire 4 to 5 million bases that are being copied in the E. coli genome. What that means is that between the 2 progeny, this one has this mutation where it is a C here, whereas in the original genotype this was a G. So, this is a mutation that has happened, and this happens because DNA polymerase has a finite efficiency, and by efficiency we want to quantify that what are the rates at which DNA polymerase is making error while it is copying DNA. So this is an example of mutation, and this type of a mutation where a nucleotide is simply written as NTD, changes. In this case, the change is from G to a C, is called single nucleotide polymorphism. And we will study loads of examples in experimental evolution where such kind of mutations facilitate adaptation and evolution. And this in short is simply written as SNPs, and they are pronounced as SNPs. So, whenever we use the term SNP, what we mean is that a single nucleotide change has happened which will lead to a change in some manifestation of the cellular of the cell. So, the question then is that this finite efficiency, what is the rate at which these mistakes are happening? What is the rate at which these mistakes happen? What you should realize that this mistake that the DNA polymerase is making is responsible for generating diversity. The more mistakes that the polymerase makes, the more different type of individuals you are going to get in the population. So, this DNA polymerase error rate is responsible for generating diversity. So, what we are going to try and do now is try and understand that in the flask example where the population went from N0 to NT, what is the sort of diversity that is generated purely because of these errors taking place because of DNA polymerase not working at 100% efficiency. All right. So, in E. coli, the polymerase makes an error the error rate in the polymerase is of the order of 10 to the power minus 3 per cell per generation. What that means is that if I have an individual cell, let us say I have an individual cell, this divides to give 2 progeny, and each one has the DNA, 1 copy of DNA. Then this number of course is just 0.001, this is roughly equal to this 0.001. What this means is that there is a 0.999 chance that progeny's DNA is exactly the same as the parent DNA. And remember, each of these divisions involves copying 4 to 5 into 10 power 6 nucleotides. We can let's just call it 5 into 10 power 6 nucleotides that have to be copied for one cell division to take place. And even with copying that number of nucleotides, the chance that the progeny DNA is going to be exactly the same as parent DNA is almost 0.999. So, this is obviously equal to 99.9% chance that there is no error. So, DNA polymerase is a machine that operates with a very high efficiency. However, there is this 0.001 or 0.1% chance that error is made. And if that happens, what that means is that progeny DNA is not the same as the parent DNA. One of the individuals is carrying a mutation. And this error rate has a symbol in all of evolutionary biology, and that is mu. So, mu is the mutation rate or the error rate associated with DNA polymerase. Hope that makes sense. So, it seems like DNA replication is an extremely faithful process. There are hardly any errors that take place. Even in this 0.1% cases where errors are made, the error is simply one error. So, this might be a G hair and which might change to a T hair and so on and so forth. So, errors are extremely rare when it comes to DNA polymerase working in an organism. The way we are going to look at this, the way we are going to look at these numbers is the following, that instead of looking at this in probability terms, we will see, we will look at it in a slightly different context, that of the 1000 divisions taking place in any flask, test tube, pond, any environmental context, we can divide these 1000 into 2. In 999 such cell divisions, there is no error. In one, however, there is an error. And this has interesting implications, as we will see in the next slide. So, let us see how big or small this number is by going back to our flask example of population going from N0 to NT. So, to make sort of our analysis a bit easier, let us just imagine that when we started the experiment, there was only one individual. So, N0 was equal to 1. We let it go for a long time, and NT is very large. So, this is long time. 24 hours is long enough time that all the media is just completely used up of the carbon and other resources that are present, and it just cells with no additional resources to further increase in number of viable cells present in this flask. So, one such popular media that is used for E. coli is called LB growth media, and this, when E. coli is grown in LB at 37 degrees Celsius with shaking, these are conditions which are very favorable for growth of bacteria, and typical densities that the bacterial population can reach in this under these conditions is 10 to the power 9 cells per milliliter of this flask. So, let us imagine that the liquid volume that we started this culture was 100 mLs, and at the beginning of the experiment there was one individual only in this flask. So, at the end of this long time that we are letting growth take place, the number of individuals in the flask is simply equal to the volume of flask times number of individuals per volume. So, we know that the number of that the volume of the liquid culture that we have that we have provided the bacterial population to grow into is 100 mils, and we also know that each one of these 100 mils supports 10 to the power 9 bacteria. So, this is 10 to the power 9 individuals per ml. And when we multiply this cancels out and we get that the number of individuals here is 10 to the power 11. So, that is the size of the population that we have at the end of this period of growth. Now, we should look back that when we look at these numbers, how many cell divisions took place? How many cell divisions? And we know this from before that if NT is the final number of individuals, N naught is the starting number of individuals, and number of divisions is just NT minus N naught, which is just equal to 10 to the power 11 minus 1. And if you do this subtraction, you should convince yourself that this is basically 1 followed by 11 zeros, 6, 7, 8, 9, 10, 11 minus 1. So, what you get is 9, 9, 9 all the way to 9. This is simply equal to 0.999 going all the way into 10 power 11. Now, for all practical purposes, this is essentially equal to 10 to the power 11. This number is just 10 to the power 11 minus 1, which is this to be exact, but we can easily approximate it as just simply 10 power 11. So, that is the number of cell divisions that took place in this flask, but we know that error is made and one error is made every 1000 divisions. Every 1000 divisions 999 are accurate and one error is made. So, one error is made every 10 power minus 3 divisions. The number of divisions that have taken place is this many. So, what you should realize, if we were to quantify the number of errors that are made in this flask, is simply equal to error rate times number of divisions, which is equal to 10 power minus 3 into 10 power 11, which is just equal to 10 to the power 8. So, in this one flask that you grew overnight, there are 10 power 3 mutations that took place. This is the number of mutations that happened in the flask. What you should realize at this point is that this is an extremely large number. Remember that the genome of the organism, this individual organism and its DNA, the DNA itself is only of the size 5 into 10 power 6. At every one of these 5 into 10 power 6 positions, there are only 3 mutations that are possible. The mutations that are possible are that if there is a, it can mutate to T, it can mutate to G, or it can mutate to C. 999 times it will stay as A. So, these are the only 3 mutations that are possible at every one of these 5 into 10 power 6 positions. That means the total number of mutations that are possible is simply equal to 3 into 5 into 10 power 6. This 5 into 10 power 6 is the size of the genome, and this 3 is the number of mutations possible at any one point. So, you could be any one of the 4 nucleotides, and you could acquire a mutation and become any one of the 3 remaining nucleotides. So, this is equal to 1.5 into 10 power 7. This is the total number of SNPs which are possible. Total number of SNPs possible. The number of mutations that happened in that one flask that we grew in lab is much greater, is approximately 7 times greater than the total number of mutations that are possible in an E. coli genome. And we will continue our discussion about the implications of this in the next video.